Hi, so this is my membrane video, my concept map. I turned the brightness down so you can see, hopefully you can see all that. So I'm going to start off with the plasma membrane. And this is a semi-permeable membrane that encloses the cytoplasm of the cell. It's the metal, so I feel like it was the most important. And what this looks like is there's two models, the David and Daniele model and the Singer and Nicholson model. And basically what the David and Daniele model says is just that proteins are on the outside. I don't know if you can read that, but it says membrane is made of a phospholipid bilayer with proteins on the outside, but this was actually seen as inaccurate. So there's a more recent model called the Singer and Nicholson model. And basically it's the same thing, it's just that proteins go through the bilayer instead. And so then if we go back to the plasma membrane, you can see, if I'm going the right way, they're made of proteins and a phospholipid bilayer. The phospholipid bilayer is pretty important. It's basically two layers of like lipids that are just help a membrane for the structure. And then the phospholipid bilayer is made up of hydrophobic tails and hydrophilic heads. And basically this is just like the tails is just the bottom of a phospholipid, and it's made of saturated fatty acids. And this is also right here, so saturated fatty acid and also unsaturated fatty acids. I'm sorry, I don't know if you can see. So yeah, they're made of this and that. And then yes, and the hydrophilic heads, well, I guess I should explain what saturated and unsaturated fatty acids are. Basically, saturated is when the carbon chain of the acid can't accept another hydrogen atom. And then the unsaturated is where the carbon end of the acid can accept another hydrogen. And it's like bent. It looks a little like kind of like a broken leg, if that makes any sense. And then let's see. So the phospholipid bilayer, if we go over here, it's amphipathic. So when a molecule is both hydrophobic and hydrophilic, and this is a property of the bilayer. Then if we kind of go over here to the glycolipids, this is a lipid with a carbohydrate on it, right here. I'm sorry, I don't know if you can see them all over the place. But yeah, this is just a lipid with a carbohydrate on it, and it is part of the membrane. So then if we kind of go back to proteins, these are diverse organic components made from one or more folded polypeptide chains, and proteins are pretty important just for everyday things like DNA and synthesis and just like energy and eukaryotic cells and things like that. So with these proteins, there's things called peripheral proteins and integral proteins, and this is a protein that only attaches temporarily to the membrane, and this attaches permanently to the membrane. Excuse me. And then if we go down to membrane proteins, excuse me. This is just a protein that is attached to or into a membrane. And then from there we can go from the membrane proteins, we can go to, I guess we can go to passive diffusion, if you can see. Passive diffusion. So passive diffusion is when like a substance is transported across a membrane without the use of energy. And I don't think I put this on here, but this can also be tied to membrane channel, and that's just proteins that facilitate this passive diffusion. I didn't put it on here, but it would definitely help with that. And then if you kind of like keep going down, we can go to the channel mediated passive transport. And that's basically when materials passively diffuse at the membrane channel. So, well, like through. It diffuses through a membrane channel. And so then if we kind of go back, we can go to the ion channel. And it's going to be right here. Still don't know if you can see the ion channel. The ion channel is basically a protein complex that penetrates the membrane to create a channel, and this can lead to the gated, right here, the gated ion channel. If you follow that arrow, it's not really connected, but the gated ion channel right here. Basically, this is just a transmembrane protein that allows ions to pass under specific conditions. Then if we continue, it just gets into like the different types of proteins that there are, or yeah, just the different type of proteins that there are. If we bring it down here, well, if we go back, so we were here, so we kind of stopped here, and I'm gonna go back to the membrane proteins and just bring it all the way down to the anchor membrane proteins, and then the transporter membrane proteins. Basically, this just helps orientate the cell toward a substrate. And then the transporter protein actively transports matter and materials across the plasma membrane. So the transport membrane, that's its job. Then we go down to the receptor membrane proteins, and these are complexes on the outside of the membrane that materials attached to interact with the cell. And then if we keep going down, we have pretty much the enzyme membrane proteins, and these are found in or on the membrane itself. These are also connected to these. That's why I kind of put an and right here. These are connected to the monolayer associated membrane proteins, and then the lipid linked membrane proteins, and then the protein attached membrane. Basically, what this is just the monolayer associated proteins. They're only associated with one layer of the phospholipid bilayer, and then the lipid link membrane associated proteins right here are associated with a specific phospholipid in the bilayer, and then the last one, the protein attached membrane associated protein right here. It's associated with a specific protein on the cell membrane too. So these are kind of all related as it ties back to the membrane proteins and just like the plasma membrane in general. Let's see, that's like the end of that. So if we go back to the plasma membrane, and I think we should go to the concentration gradient. No, actually we're not done. I forgot something. So go back to the where did I put it? To the membrane proteins. There's also something called detergent molecules. These molecules, they purify, isolate, renature, and cellularize membrane proteins, and they kind of like break them down as well, kind of like detergent, like in soap. So there's that. And then after we're done with that, we can finally actually go back to right here, which is going to be the concentration gradient. So the concentration gradient is basically like the gradual change of the concentration of a solute in a solution. And there's like different types of solutions as it pertains to the concentration gradient. So there's going to be, I don't know if you can see it, but isotonic solution, hypotonic, and hypertonic solutions. Basically, so like the isotonic is when the environment has like equal amounts of solute. Hypo, I said that's isotonic. So hypo is going to be that it has more, and then hypo means less, and hyper means more. Then we can kind of go back to the electrochemical gradient, and that's just a measure of ions' ability to diffuse through the cell membrane. And then there's different ways that they can do this, and that's through osmosis or active transport. And osmosis is when materials pass through like a semi-permeable layer from an area of low concentration to an area of high concentration. And then they also do this through active transport. And this is when enzymes move materials like themselves from areas of high concentration to areas of low concentration. And it's not on here, but what would actually really be a part of this electrochemical gradient would also be simple diffusion. And this just means like particles from an area of high concentration to an area of low concentration. So basically just the opposite. Yeah, so basically just the opposite of osmosis and active transport, but they're very similar. And like I said, like the hypertonic and hypotonic and hypertonic solutions is basically just when like the environment has higher levels of solute than the cell does. So hypertonic would be more, it has more, and then hypotonic, like I said, is when the environment has lower levels of solute than the cell does. And then after that, we can kind of conclude with the going back to the membrane, plasma membrane, and kind of going to selective permeability. And selective permeability is pretty important because it only like allows specific particles to pass through the membrane. And then vesicles I kind of added because they're made of at least one layer of phospholipid bilayer in the cavity, made from membrane material used to transport material into and out of the cell. So they're pretty important with like transportation and like just like I said, selective permeability, what comes in and out of the cell, what's allowed in and out of the cell. And this is a property of plasma membranes. So yeah, that's it. I don't know if you can see that.